In this last video, we will talk about histology of both the male and female reproductive tracts. This is another area where we could spend a whole semester just looking at histology of this system. However, on the lab exam, there's really just a few slides I want you to focus on for preparation. So we'll start with the male system and then we'll look briefly at a slide of the ovaries and spend some time identifying the different stages of oocyte maturation. To start, we're going to look at the epididymis. So as a reminder, this is that convoluted tube that is wrapping around the testes. So this is the site of sperm maturation and storage. Full features of the epididymis are this repeating pattern of tubes. The epididymis itself looks like a really long, highly coiled tube. What we have done is taken a cross section of the epididymis, and now we're looking down on the lumen of different tubes here. Around the lumen, we're going to have a layer of smooth muscle, which is nicely labeled by our Yale histology site. That's going to be the outermost layer. Inside, this should look pretty familiar. So we're going to have a layer of columnar epithelium. And then one of the really interesting features of the epididymis is the presence of stereocilia, which we've seen once before. We saw them in the ear, and we see them again here in the reproductive tract. So stereocilia are like hair-like cells. Cilia have a motor function, so these are going to help transport or propel sperm. So they have that mobile function. That is what I want you to use to identify the epididymis. This is really the only structure in this male reproductive tract that has stereocilia. So if you see this repeating pattern of tubes, look for the lumen and try to identify whether or not the tissue has stereocilia. If it does, you know you're dealing with the epididymis. Next up, we have the testes, and I did want to incorporate some examples from our modules, so this slide is available to you. A lot of students get mixed up when they're trying to identify testes versus epididymis because we have another pattern of repeating tubes. And in this case, the term for these many tubes is seminiferous tubule, little tube. So this is going to be the site of sperm production. If we look closer on these slides, we will see a couple types of cells. So we're going to refer back to our Yale histology site. I like this one a lot for reviewing tissue of the reproductive system, and that's how I would recommend studying for Lab Exam 3. So let's find our seminiferous tubules. What we've done is we've taken this view and zoomed in just a little bit so that we can see the types of cells that we would find in the testes. We're going to see lots and lots of epithelial cells just lining the lumen of these many seminiferous tubules. There are a couple types of specialized cells, and so one of them is pointed out on this slide. Between the tubes, there's going to be a little space, and usually it is darker stained. This is where our Leydig cells are hanging out. So they will be a little larger, usually they'll have a dark purple nucleus, and they fit in between the spaces between the tubes. These Leydig cells are producing testosterone. We have a couple other types. We will find Sertoli cells. Those are going to be scattered kind of within this epithelium here. Another name for the Sertoli cell is nurse cell. So these are going to be nursing or nourishing sperm cells during that production process. So we're not going to pick out individual nurse cells on this slide, but I want you to recognize we would find them in this epithelial layer. And we also have myoid cells. We would find myoid cells along the outer border of each of these seminiferous tubules. So they're really thin, and if we were able to zoom in, we would see that these kind of resemble smooth muscle. And that makes sense based on the name. If you see myoid, you might think that sounds a lot like muscle. These cells are contractile, so they're actually going to contract and help compress the tubules here and help transport sperm out of the testes into the epididymis. So 3 types of cells, all with slightly different functions. Our PowerPoint has a really nice slide pointing out all of those cells, so I recommend taking a look at that. But I can tell you now, the lab exam is not going to be that detailed. I want you to be able to identify testes versus epididymis, and I want you to understand that the tubular unit of the testes is going to be a seminiferous tubule. You don't need to know cells, but you do need to know tubules. Last up for the male reproductive tract, we have sperm. Sperm are very tiny. You can zoom into 40x magnification with our microscopes in class and still have trouble finding them. So this might be a really good PowerPoint question, for example. Sperm have a head and a tail. The head is going to be the round main body of the sperm, and then this really long tail is a flagella that is going to help propel the sperm through fluid. It helps them to swim towards an egg. So pretty basic, no head and tail, and recognize that the tail is going to have a flagella, and that's all you need to know for sperm. Let's switch gears now and talk about the female reproductive tract. So once again, the Yale site will be very helpful for reviewing for this. One other thing I want to point out on the Yale site that you may not know about is that the Yale site has a virtual microscope. So while looking at these types of images, labeled or unlabeled, is very helpful. If you want something more like our practice in class with the microscopes, all you need to do is navigate to a specific slide, and then you'll scroll down past the reading, past the pre-lab and the slides, and then we get these virtual microscope slides. If you click on those, it's going to take you to a virtual microscope, which you can zoom in or out with. This one is labeled, so there may be additional features on the slide that are beyond the scope of this class. So you don't need to know all of the markers here, but if you want to practice, you can slide over to a blank microscope slide, and then you are free to click around and explore. So this is another really helpful way to test what information you can recall. I bring that up because the ovaries are pretty complicated. Structurally, the ovaries are going to have an outer cortex and an inner medulla, very similar to several other organs that we've talked about. In this case, the cortex here is a little darker stained, and then the medulla is going to be pretty busy with blood vessels. So you may see those lumen as you poke around this slide. Today we are concerned with the cortex. All of this stuff around here. This is where the action of producing follicles is going to happen. So let's talk about this process of how we produce sex cells, in this case, oocytes. So there's a term for this called oogenesis. That is the production of oocytes with the eventual end product being a mature egg or ova, ovum for singular. All of that is happening within these round units called ovarian follicles. So that's going to be the site of oocyte maturation. These follicles are also producing estrogen, and they're going to help regulate the menstrual cycle. They're going to be lined by some specialized cells that are producing those sex hormones, estrogen and progesterone, and those cells are also assisting with oocyte development. Their counterpart in the male system would be the nurse cells, which are nourishing sperm. Again, the end product is a mature egg, and that is going to be released into the fallopian tube. Before we try to identify the different stages of follicle development, I want to refer back to one of our PowerPoint slides. And I think this does a really good job of showing the entire process of oogenesis. So once again, the end product is a mature egg, but there are a lot of steps that have to happen to get to that point. You may have seen this type of schematic if you've taken a biology course before. What it shows is the process of mitosis, regular old cell division, and then meiosis, that process with the end product being a mature sex cell. There's going to be a bit of mitosis that happens, a bit of meiosis that happens all pre-birth, with the result being the production of a primary oocyte. So this is that egg, so to speak, that is going to hang out in the ovaries before birth. That's part of the reason why our number of eggs is determined before we are even born. It's because these primary oocytes are produced, and then their development is going to be arrested at prophase until puberty. So these are what we are referring to as that pre-birth egg count. Once that puberty is triggered, the process is going to complete. So we're going to have a primary oocyte turning into a secondary oocyte and eventually into a mature egg. So while you don't need to understand this diagram completely, I want you to start getting comfortable with these terms: primary oocyte, secondary oocyte, and ovum. We're going to flip back and forth between our Yale site and our module slides for the next couple of minutes. The default state for these follicles before anything happens to them at puberty is they're going to hang out as a primordial follicle. So if we zoom in here, we're going to see these very small round units. And there are lots of them towards the outermost part of that cortex. These are those primordial follicles. You can think of this as the default or rest mode for all follicles in the ovary. So this is going to be that pre-birth status. These are what is contributing to that egg count. Within each of those follicles, we're going to have a primary oocyte. And so that's just this little round cluster of cells. At this point, it is really small because there hasn't been that subsequent meiosis. Histologically, what you'll see is just a simple layer of squamous epithelium, just one layer of really thin cells outlining each of these follicles. While I'm not going to test you on the epithelium here, I want you to keep that in mind because that shape and the number of layers is going to change as these follicles grow. So from the primordial follicle, we're going to get this structure called a primary follicle. And our site has some really good examples of these primary follicles. This was primordial with that single layer of squamous epithelium. Next stage is a primary follicle. So what's happened is that layer of squamous epithelium has now expanded and we get this more cuboidal epithelium. And you can see that there's a couple layers deep here. So this is our primary follicle. At this point, we still call the middle structure a primary oocyte. From here, from primary, we go to secondary follicle. And all that's really happened is this layer of cells, this layer of epithelium has gotten much bigger. So we have many more layers of cuboidal epithelium now. We still have that primary oocyte. One additional thing to note is we have this layer starting to form around the entire follicle. And this is called a theca, T-H-E-C-A. Don't worry about identifying that for the lab exam, but we're going to keep our eye on that and see how it changes into the next stage of development. So the last and most mature follicle is going to be a tertiary or Graafian follicle. So you'll see Graafian. Either term is acceptable on the lab exam. Think of this as the mature version of an ovarian follicle. At this point, it looks a lot different. This follicle has gotten way bigger. We now have this really big space in the middle that is called an antrum. This is going to be filled with follicular fluid, and the oocyte also looks different. In this case, once we reach this mature version of the follicle, we call the oocyte a secondary oocyte. It has now matured and it's ready to be released to the fallopian tubes. So during normal ovulation, this follicle is going to rupture to release that secondary oocyte. So this will break apart. Secondary oocyte will be swept up by fimbriae within the fallopian tube, and then it'll begin that journey to the uterus. Post-ovulation, this structure will reorganize. It will reconstruct itself into a structure called a corpus luteum. At that point, it's going to secrete progesterone, and that is going to signal those changes within the structure. And if no pregnancy occurs, then this is just going to go away. It's going to shrink down, and the process will begin anew with a different follicle. Putting that all together, let's go to our human protein atlas. This is another great way to practice with the unlabeled slides. Once again, we have the entire ovary. We're going to have a really busy medulla full of vascular structures. Cortex is going to be where we have the most activity. So this slide will give you primordial follicles, that default state with just a simple squamous layer. It also has primary follicles. So you can see that change. We went from a simple squamous layer containing a primary oocyte, and then that simple squamous layer becomes cuboidal. So it's starting to expand. And eventually we'll get to this phase where the expansion is complete. We have lots and lots of cells surrounding that big antrum, and we have a secondary oocyte. I want you to understand those three different phases, primary, secondary, and tertiary follicle, as well as the default state. I want you to recognize that these follicles are primordial. until something happens to them at puberty or later. They're going to hang out in this default state. That is it for histology. So there are going to be parts of the PowerPoint that I'm not covering in these videos, mainly because they're not emphasized on the lab exam. Things like the layers of the uterus will not be emphasized: perimetrium, myometrium, and endometrium. I will also not ask questions about the menstrual cycle. However, those are all fair game for lecture, so I recommend taking a look at the PowerPoint slides. There's a summary of the stages of the menstrual cycle, what is happening in each phase, and how long each of those phases lasts. Please take a look at that. I think that will help you with lecture. There's a couple helpful diagrams in there about how the uterus lining, the endometrium, is going to change with the different phases of the menstrual cycle. So that is all good information to know for lecture, but stuff that will not be tested on the lab exam. Good luck studying. I know everyone has put in a lot of work this semester, and hopefully it will pay off on that last lab exam.